Hello class, welcome back to genetics. Don't forget last week we talked about genotypes versus phenotypes, and then also dominant versus recessive alleles. Remember that alleles are just different versions of a gene. So here's an example of a homozygous dominant genotype, a heterozygous genotype, and a homozygous recessive genotype, and then their corresponding phenotypes. If you need more practice with that or think you need to go back and review, there was the worksheet that you finished up with on Friday, or you can go here for more practice. This is in your packet, and this is looking at your different traits. Each one of these is a dominant trait, so you just need to look at if that's something you have or not, and then think of what your phenotype would be in that case and your possible genotypes for that then. If you're good though, go ahead and start today's lecture. So, we are talking about Gregor Mendel. He is the father of genetics. He's an Austrian monk. And in order to come up with all of his evidence for genetics and what exactly was going on, he experimented with pea plants. Very exciting. So what he figured out was that there was something that carried traits from one generation to the next. He didn't know what that was. They had no idea what DNA was at the time, but from all of the breeding he did with the plants, he knew there was something going on. So he actually studied physics and botany at the University of Vienna, and since he was a monk, he had plenty of time to garden and experiment with these pea plants. He actually worked with them for over 8 years, and in that time tested over 28,000 pea plants. So here are some examples of phenotypes that he tested for. There was the seed coat color, as well as the flower color we talked about. The seed color there, the seed shape, whether it was wrinkled or smooth, pod color, pod shape, the stem height, and also the position of the flower on the stem. And here is where all the magic happened. Okay, so something to understand when we're talking about genetics now are the generations. So you'll typically see a P, and that stands for the parental generation. That's the very first generation. Then you'll see an F1 generation, which is the first generation. These are the offspring of the P generation. The offspring of the F1 generation are the F2 generation, and this will actually typically be a cross between two F1 individuals. So here's an example of a P generation. We're going to cross a purple flower with a white flower. And something else here, these are called true breeding, because both of these are going to be a homozygous genotype. Whenever he crossed these flowers, you saw it in the F1, they were also going to have purple flowers, and these are considered hybrid because they would have the heterozygous genotype. So then when he crossed 2 from that F1 generation, this is what he ended up with, a 3-to-1 ratio. Okay, so let's talk about how we can figure that information out and how we know what exactly we're going to get. So in order to do this, we use something called a Punnett Square, and this is a tool we use for predicting the possible allele combinations in gametes, which will tell us the traits of an offspring. Okay, so don't forget that gametes are our egg and sperm, and those are the cells we're using for reproduction, and that's really what we're talking about here. So the letters we're using, those uppercase and lowercase letters, are symbols for the genes. And the genes will always exist in pairs because they're found on homologous chromosomes. Okay, so here is an example. So in this example, we're going to cross our homozygous dominant purple flower with our homozygous recessive white flower. So this is going back to that example we just ran through. On your Punnett square, you're going to take one parent's genotype and put it on one side, and the other parent place that on the other side, like this. Then all you'll do is carry down and over. Okay, so you bring this dominant allele down and this recessive allele across, and this is the genotype that you end up with. So keep in mind that this separation here, putting this dominant allele over this box and this dominant allele over this box, that represents meiosis and the splitting up of our homologous chromosomes. This right here, where we combine an allele from each parent, this would be fertilization. Okay, so back to the Punnett square. This is the genotype we end up with in this box. Since there's a big B all across the top, a little b all across the side, every single offspring here would have this genotype. Okay, so our genotypic offspring, we won't have any with the homozygous dominant genotype. All four of them will have that heterozygous genotype, and none of them will be homozygous recessive. So now let's look at what happens if we take two from this generation, and cross them together. Okay, so this is what we're working with. Both are going to be purple phenotypes with a heterozygous genotype. Okay, so you pull down and across, and in this offspring would have a homozygous dominant genotype. So we'll continue that. We'll pull down and across, and that's what you end up with. Go up here, pull down and across. Keep in mind you always want to put the dominant allele first, and then the very last one would look like that. So now for our genotypic ratio, there's one that is homozygous dominant, two that are heterozygous, and one that is homozygous recessive. So this is where we get our 3-to-1 ratio of 3 purple flowers to 1 white flower. Okay, so let's talk about a couple laws really quick before you move on. So a law of dominance that Mendel came up with. This is talking about how we have dominant alleles. So a cross between a homozygous recessive and a homozygous dominant means that one version will essentially disappear. That's referring to the recessive trait. Okay, so it's going to get masked by that dominant one. The law of segregation is what I was just explaining about meiosis. So two alleles for each trait separate when gametes form, and that happens in meiosis. Okay, so that is shown right here. So parents pass only one allele for each trait to each offspring, and the Punnett squares demonstrate this. Okay, so if this offspring were possible, it would be coming from this one allele of that one parent and this one allele of the other parent. So here on the side and across the top, that's because of meiosis. Here in the middle, that's your fertilization. Okay, then the law of independent assortment. So genes for different traits are inherited independently of one another. Okay, so that's just saying that genes are not dependent on one another. Okay, so your hair color is not going to be inherited in relation to your eye color. Those are separate things. Okay, and this is just a little example to show you how we know that that's true. All right, so now what you can do, what I would like you to do is, well, first of all, yes, I am enjoying Lumen, thank you. Okay, so first of all, you do have this worksheet. It's not a worksheet, it's a guide in your packet if you're still not quite sure about the Punnett squares. But what I would like you to do is find this worksheet in your packet that says Punnett squares and complete these practice problems. There are 7 of them here that I would like you to do. And that is everything. Okay? Let me know if you have any questions. Just email me. Thanks.